Hello everyone, welcome to my channel The Learning Tree. Today's topic will be asexual reproduction from the chapter How Do Organisms Reproduce. So before starting we should know what is asexual reproduction. So the type of reproduction in which only one parent is involved and where there are no fusion of gametes, that type of reproduction is called as asexual reproduction. So in sexual reproduction, what happens is two parents are involved or the fusion of gametes takes place, whereas in asexual reproduction only one parent is involved and there is no involvement of any gametes, and the young ones that are being produced, so the next generation that is being produced, they are almost identical to the parent cell because there is no fusion of gametes. So that is why identical young ones are produced, and it is a faster method of reproduction. This asexual reproduction occurs both in unicellular as well as multicellular organisms. Now, what are the different types of asexual reproduction? So if you see here, so we have fission. Fission can be two types. One is binary, another is multiple fission. Then we have budding, fragmentation, sporulation, and vegetative propagation. Again, vegetative propagation is of two types. One is natural vegetative propagation and the second one is artificial vegetative propagation. The one which is done by humans or by the intervention of human beings, so that comes under artificial. Naturally the plants or the organisms will undergo vegetative propagation. So under natural we have propagation through root, stem and leaf, which are the vegetative parts of the plant, and artificial propagation involves cutting, grafting, layering and tissue culture. So we will study each of these types one by one. So let's begin with the different types of asexual reproduction in unicellular organisms. The first one is binary fission. Okay, so binary fission mostly occurs in unicellular organisms like bacteria and protozoan like amoeba, paramecium. Okay, so here what happens in binary means two. Okay, that way you can remember two. That means two daughter cells are formed from the parent cell. In this binary fission, what happens is the parent cell, first the nucleus will divide and then the cytoplasm will divide, and ultimately two daughter cells are produced from one parent cell. So this type of reproduction or asexual reproduction is called as binary fission. Binary fission is also seen in leishmania. So one thing we need to remember about leishmania is in binary fission like amoeba, there is no specific site where the binary fission will take place, whereas in leishmania it occurs in a specific position or in a definite orientation in that particular organism. Otherwise, in case of amoeba, it can occur anywhere inside the cell of the amoeba. So that's what happens these are the different stages in binary fission. So this is a parent cell. So you can see the two nucleus divides and even the cytoplasm starts dividing. Okay, and then two daughter cells are separated and each of these daughter cells are two amoeba cells are produced. Okay, that's how binary fission takes place in amoeba. So you can see here, so this is the parent amoeba. So first the nucleus starts elongating and divide, and even the cytoplasm starts dividing, and then two daughter amoeba cells are produced. And if you see on the right hand side, this is the actual amoeba that you can see under microscope. It shows how the binary fission is taking place in amoeba. So you can see here the two daughter cells are being produced. This is the parent one, this is the two daughter cells being produced. Now coming to paramecium, it also undergoes binary fission. So this is how a paramecium cell looks like. Okay, so here you can see, so first the nucleus will divide. Okay, the nucleus starts dividing and then the cytoplasm also starts dividing, and then two daughter parameciums are formed. Next, leishmania. Leishmania is the organism that causes color azar. Okay, the disease called kala azar. Okay, that's caused by leishmania. So leishmania, this is how a leishmania looks like. So in leishmania, if you see, longitudinal division occurs. So you see here the nucleus, can you see the nucleus? So here the nucleus starts dividing into two, and then if you see here through the longitudinal axis, the parent leishmania divides into two, and then these two will separate itself, and then two daughter leishmania will be produced through binary fission. So this is about binary fission. Next comes to multiple fission. Multiple means many. So many daughter cells are produced from a single parent cell. So this is seen in commonly in Plasmodium. Okay, so Plasmodium is a malarial parasite. So here if you see, so in Plasmodium what happens during unfavorable conditions. Now this is the Plasmodium. Okay, so that causes malaria. So Plasmodium, what happens is during unfavorable conditions, it forms a cyst. Okay, protective covering outside its cell called a cyst. Okay, so what happens within the cyst, what happens, the cytoplasm starts dividing, and then many cells are formed inside one parent, inside the parent Plasmodium cell. So and once the situations or the environment becomes favorable for the Plasmodium, that particular cyst will break open, and then all the leishmania cells, daughter leishmania cells will be the Plasmodium cells will be released. So this is the one cell, particular cell. If you see here, so a lot of leishmania cells are produced inside one cell. So that will, then once this condition becomes favorable, you see here the walls gets ruptured, and then all the leishmania cells are released. So they are called, the daughter ones are called as merozoites. Okay, and this cell, single cell containing many nucleus, that is called as schizoid. So in Plasmodium, so the schizoid cell, first the division will occur, nucleus division takes place, and then many, even a cytoplasm starts dividing, and many Plasmodium cells will be present inside the parent cell, and once it ruptures, the cell wall ruptures, all the leishmania of daughter cells will be released, which are termed as merozoites. So this is about multiple fission in Plasmodium. Next comes budding, which is also a type of asexual reproduction seen in yeast and hydra. Okay, yeast is a type of fungus and also seen in hydra. So in yeast, what happens, the parent yeast cell will develop a small outgrowth at its upper end. Okay, and then the nucleus will divide and move towards, one of the nucleus will move towards the developing bud, and then slowly what will happen, the cytoplasm will divide, and this part of the bud part will separate as a matured daughter yeast cell, and then the parent one will remain as it is. So that is called budding. Budding means bud. A small bud or outgrowth is developed in case of yeast cell. Okay, so here the first nucleus will divide, and then one of the nuclei will move towards the daughter cell, and then the daughter cell separates from the parent cell, forming a new daughter cell or forming a new yeast cell. So yeast, it can either be one single bud or it can be multiple buds in the form of chain. So that is also seen when conditions are favorable. In yeast, you can see multiple buds are being formed in a single parent cell. In this case, this is single budding occurs. The one single bud was formed and it was got detached. We can also see multiple budding in yeast also. So in the second picture, if you see, it's the same thing. So the nucleus starts dividing, cytoplasm divides, and now you can see all the yeast, daughter's yeast cells are formed in the form of chain. Okay, so that is also seen in yeast. Next we see budding in hydra. Even in hydra also buds are developed at the lower end. Okay, so here the lower end of the hydra you will see the parent hydra, you see a smaller outgrowth is seen, slowly it grows. Okay, so here if you see the small hydra is growing, and then what will happen, once this small the new bud that is being developed is matured enough, it will get detached, and the daughter hydra will again develop into a complete new organism or a new hydra daughter cell. So that's about budding in hydras, which is also a type of asexual reproduction. Now this hydra, the daughter hydra, it detaches itself and it can grow independently. Next we'll go to the multicellular organisms, the type of asexual reproduction in multicellular organisms. So under that, first is your fragmentation. So in fragmentation, what happens, a single organisms will break down into small fragments. Okay, so each of these fragment will give rise to the new organism. So that is called fragmentation, where an organism will have will divide into various fragments, and each fragment of this organism will grow into a complete new organism of the same species. This is mostly seen in Spirogyra. So Spirogyra has a filamentous body. Now if you see here in the picture on the right hand side, you see this, that's how Spirogyra looks like under a microscope. So if you see, you can see the filamental structure. Okay, so each of these lines, can you see, these are called as septa. Okay, so this, so if you break in any part, suppose this fragment, if you're breaking it down, this one, so different fragments, if you cut it open, then each of these fragments will give rise to one Spirogyra. Okay, that's how, that is called as fragmentation. Next comes regeneration. Regeneration means to regenerate, to produce new ones. Okay, you must have seen in lizards, you see the tail of the lizard, if it breaks down, what will happen after a few days? You see the lizard, same lizard, in the same lizard the tail grows back. So that is called regeneration. Okay, same goes for starfish. If you cut a piece of the starfish again will start regrowing in that particular part which is cut. So that is called as regeneration. Now in regeneration also is a type of asexual reproduction where specialized cells are there which will get involved in reproductive part. Not all organisms undergo regeneration. Specific ones which have specialized cells for reproduction, those will only undergo regeneration and produce new organisms. In case of lizard and starfish, although they are producing producing new parts, but then it is not growing into a new complete, the tail itself is not growing into a new lizard. So same, but there are certain organisms where the one specific part will give rise to new individuals. So that comes under, that will happen only when there are specialized cells. Okay, so if you see here, so that is common in planaria. Planaria is an organism where regeneration occurs. So that mode of asexual reproduction is seen in planaria. So this is like a flatworm. If you see the picture, so that's planaria. So if you see, if you cut the planaria, okay, suppose you cut it into two halves, you see each of the fragments that you get. So these three parts, so each one will grow into new planaria, because they are having specialized cells which can undergo reproduction, asexual reproduction, to produce new planaria. But this does not happen in the lizard. Only the parent lizard whose tail was cut, that parent will grow its tail, but from the tail we will not get the lizard. So that is that's a different type of regeneration. But in case of planaria, we can see individual fragments or individual parts will give rise to new planaria. Okay, now another one is the spore formation. That is another type of asexual reproduction which takes place through spores. So the spores will be the reproductive part of which will take part in reproduction. So in spores is common in rhizopus, that is a fungus. We can see, we must have seen the braids, like braid molds is commonly called as braid mold. Okay, so if you keep a piece of braid outside for a long period of time, you can see the fungus growth over there. So these are nothing but rhizopus. Okay, so these rhizopus undergo spore formation and multiply through asexual reproduction, that is a spore formation. Now what happens is, in rhizopus have a it has a thread-like structures called hyphae. Okay, so if you see in the picture on the right hand side below, you see these are the hyphaes. Okay, so these hyphaes will give rise to stalk-like structures called as sporangiophore, and each sporangiophore bears sporangium. Okay, this sporangium contains the pores. The spores that are present, which is involved in asexual reproduction, is present inside the sporangium. So this is mostly also happens during unfavorable conditions. So that's the protective covering. Now once the condition becomes favorable, the sporangium will burst open and the spores are released. And each of these spores, wherever it will fall, it will give rise to new rhizopus, or the fungus will grow separately. So that is the asexual reproduction through spore formation. Next we have vegetative propagation. In plants we have one reproductive part, another is the vegetative part. Root, stem and leaves of a plant are the vegetative part, whereas the flower is the reproductive part. Now since we are talking about asexual reproduction, so that is through vegetative part of the plants, that is roots, stems and leaves. Okay, so here what happens, when we talk about stems, we have again stems, we have rhizomes, parts of the stems will, through vegetative propagation, we can have through rhizomes, bulbs, runners, tubers, etc. So these all comes under the stems. Then we have the roots and the leaves. Now, two types of vegetative propagation, as we have discussed earlier. One is through naturally, the plants will naturally undergo vegetative propagation, and second one is artificial vegetative propagation, which is done through the intervention of humans. Now, through roots, we have example as mint. So if you so mint reproduces through the roots, then sugarcane, jasmine by the stems, bryophyllum by the leaves. Okay, so if you have seen bryophyllum leaves, you can see so in the leaves you can see small buds, right? And from these buds small plants arises, and once this falls off on the ground, this will give rise to a new bryophyllum plant. Okay, so that's vegetative propagation through leaves. So these are the different vegetative propagation. This is root propagation in sweet potato. Then we have the rhizome. These are the stem ones: rhizome, tuber, okay, bulb, runner, sucker. These all are the under stems. It falls under the stems. And this is the leaf, the bryophyllum leaf, as we have discussed. So in the leaves, buds are formed and then ultimately give rise to new bryophyllum plant. Okay, so these are the different natural vegetative propagation in plants. Next, coming to the artificial vegetative propagation. So what happens here, as I've said, it is through human control methods or human intervention is there. We are growing it artificially in labs under controlled environment, or we are doing it through the help of different tools. Okay, for example, cutting is a part of artificial vegetative propagation. Layering, grafting, and micro propagation are also known as tissue culture or plant tissue culture. So these are the different methods that comes under artificial vegetative propagation. So these techniques, why do we use artificial vegetative propagation? So it is used to propagate desirable. Suppose you need a specific trait, you want to see a specific trait in a plant, so you can go for artificial vegetative propagation. You want to grow plants in a faster, so that also we can go for artificial ones. Or you want to grow plants which does not grow seeds, so that also we can do under artificial vegetative propagation. So these are the advantages of vegetative propagation. Now first is cutting. As I've said, it is a part of artificial one. So cutting is what? We cut a part of the plant and then you put it under soil or plant it in the soil, and slowly after a few days you might see the roots growing from there, and then that particular plant will again grow into it will grow bigger into a new plant. Okay, so that process is called as cutting. So you cut a piece of the stem, okay, then you cut it, and then that cut part you put it under soil, okay, and after a few days you see roots growing from that particular cut part, and then this will slowly grow into a new one. That is called, process is called cutting. Okay, so cutting mostly we see it in rose plants, where we do cutting. We cut the rose plant, stem of the rose plant, you can put it on the soil, and then you can see a new rose plant growing out of it. Next you have grafting. Okay, so in grafting what happens is you take a particular plant, cut it into half. Okay, one part of the shoot part of the upper part of the plant is then grafted into another plant which is rooted to the soil. Okay, the upper part which you have cut, that one is called as scion, and the part which is the one which is stuck to the ground, it is called as stalk. Okay, so what happens, you will both will be joined together through grafting. Okay, scion and the stalk will be joined together, and then they'll be wrapped and waxed for a short period of time. After that, you will see that both the parts are joined together, and then results in a desirable whatever traits you want to see in that particular plant. So that process is called as grafting. Okay, you can see, if you want to see multiple colors in a rose plant, you can go for grafting, where you cut a yellow color rose plant and put it in a white or a pink color rose plant. You can see multiple color rose, like white, pink, and as well as yellow color in the same rose plant. So that can be done through grafting. Okay, next is your layering. So where a plant is there and a part of the stem is bent to the ground. Okay, so for a particular stem you choose and you put it under the ground and place the soil over it. After a few days you see the adventitious roots coming out of it. Now this part, if you detach it, it will grow into a new plant. So that is called as layering. Okay, you can see, so this part is put under the soil, a part of the stem is put under the soil, and you can see the adventitious roots coming out of it, and from this part a new plant can grow. Next is the tissue culture, the plant tissue culture, the most important one. So plant tissue culture is done in controlled in a sterile condition, in specifically mostly in the lab, where there is no contamination. Here you can get like you can take any part of the stem or a tissue or a cell of a particular plant or whatever trait you want to see. So that particular plant you choose, a small portion of it you will take it and you'll put it in a nutrient medium, okay, in under aseptic conditions, okay, that is sterile condition, and that after some days or few days you will see a mass of cells growing on that particular medium. So that mass of cells is called as callus. Okay, this is undifferentiated cells, because these cells are not differentiated till now, like which part will do what, or which part of the cell will grow to root, or which part will grow into shoot. So that was not defined, okay, until the callus part. Now from this callus again it is being transferred to another nutrient medium, okay, where the required nutrients will be provided, and then this callus will differentiate, all the cells will differentiate and grow into new plants. Okay, so that technique is called as tissue culture. So if you see here, this is your callus, it's a undifferentiated mass of cells. Now from that tissue a small mass of cells grows, and then again it is transferred to a nutrient medium, where again from there the specific plants will grow. So that is comes under tissue culture. Now what are the advantages, or what is the importance of vegetative propagation, why it is so important? Now plants can bear flowers and fruits earlier. So in vegetative propagation you can reduce the timeline of a plant for bearing flowers and fruits, so it will be faster, okay, plants. And it is also helpful for plants which have lost the ability to produce viable seeds, because we are using vegetative parts of the plant, that is root, stem and leaf. So from there also we are growing plants. So it is those plants which cannot bear seeds or which cannot bear flowers, we can use those plants and also undergo vegetative propagation to get newer daughter plants, okay. And all plants are genetically almost similar to the parent cell, because since we are using the vegetative parts, so that's why it is on the new plants that we are getting, it is almost identical to the parent plant. So if you want a desired plant, same specific characteristics of or a trait of a particular plant in the next generation, you can use the through vegetative propagation, you can get the same trait or the variety. Then you have seedless varieties can be obtained. So if you don't want any plant with seeds, then you can opt for this method for seedless varieties. Then it is used by horticulturists in developing methods like layering, grafting to grow many plants like sugarcane, rose or grapes. So it can be used to cultivate sugarcane, rose, grapes based on the demand or based on the traits that you are looking for. So these are the various importance of vegetative propagation. So I hope you have understood my explanation. Thank you for watching. Keep watching my other videos. I'll be uploading soon the other chapters also. So do like, share and subscribe my channel. Thank you.